In this video we will discuss about HTPBD characterizations. So they wanted to show from these figures results that are presented in this figure that their oral CRISPR knockout methods in the colorectal cancers has a higher efficiency compared to the traditional CRISPR knockout methods. So in the panel A they shows how they prepared their HTPBD. That is, they took the PBAE and then inject this CRISPR plasmid into this PBD, and which then looks like this. Then in order to increase this efficiency towards this mucus, what they did is they connect this or integrate this PBD with HA and TMO, and as TMO is a zwitterionics that increase the efficiencies to reach to the increase the efficiencies of this uh, plasmids to reach to the mucus. So here the HTPBDs, it looks like this, and they check its transfection efficiencies and other parameters. And in terms of transfection efficiencies, we can see that its efficiencies are higher compared to the controlled either PBD alone or with this HPBD which does not have TMAO, which is the zwitterionics. When they check the endocytosis, they can see that in the endocytosis also the different HTPBDs works higher compared to the control, that is PBD or HPD alone. And in terms of the anti-fouling coefficients, it also shows that HTPBDs works higher compared to the PBDs or HPD. And when they check all these three parameters together, they can see that HTPBDs outperforms compared to the HPD. And thereafter they check the image of this PBD or HTPBD using the scanning electron microscope, and when they compare their size, they are able to see that the PBD has a slightly smaller size, around 120 nanometers, whereas HTPBDs has a slightly higher size, around 133 nanometers. And when they check the zeta and thereafter they check the zeta potentials, here we can see that HTPBDs has a low zeta potentials, indicating that its positive charges are being shielded by this HATMO, whereas in the case of PBD the zeta potentials are not neutralized. Thereafter they checked whether this PBD is able to encapsulate the plasmids altogether or not, and here they check the cellular uptakes, and they can see that the formations of the CRISPRs, indicating that this plasmid has been efficiently integrated into this their PBD system. Now they wanted to check whether how stable is their plasmid. So they react these plasmids, which is quite stable in the PBS, with the DNase1, and then check they react their plasmid and PBD and HTPBD with DNase1 and check whether they are able to survive this DNase1 treatment or not, as in the mucus there are a lot of DNase are found, and for the plasmid they can see that there is no presence of the plasmid when it's treated with DNase, and PBD has a low presence, but HTPBDs almost have negligible effect of this DNase1. And thereafter they check the effect at small intestine, as the small intestines has the largest immune cells. They wanted to show that it does not be cleared by immune cells in a short duration of time. So they react their HTPBDs with the gastric fluid, and they can see that for the plasmid they do not see any signals, but for the case of HTPBD they are able to see some signals. And similarly, when they reacted with intestinal fluid also, for the plasmid they do not see any results, but for the HTPBDs they can see the results, indicating that their HTPBDs are quite stable. And thereafter they check this HTPBDs and PBD and controls expressions. They can see that PBD has slightly higher expressions compared to the HTPBD. And next they wanted to check whether they check the expressions of these PBD and HTPBDs in different conditions, and they can see that the PBD's expressions are high in the normal case, but HTPBD's expressions are even higher in the absence of the sucrose, which is essential for its transfection to the mucous site where the colorectal cancer is there. And in terms of the transfection, here in the panel L N, they wanted to check whether their HTPBDs can be lyophilized or not. So this is the result after lyophilization, and they can see that HTPBDs are able to can be lyophilized even without the presence of sucrose. And they check the results for the transfection efficiencies for the lyophilized with sucrose and without sucrose, and they can see that even without sucrose the transfection efficiencies are higher compared to the PBDs. And in the panel P, they shows the immunofluorescence image of the HTPBD and PBDs, which shows that HTPBDs has a higher transfection efficiencies compared to the PBDs. So all these results together sum up to a point that their oral CRISPR injections method, that is HTPBDs, can be used to selectively target the mucus where colorectal cancers are found. So that's all for this video. So if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.